Hi folks, welcome to the third lecture in the Experimental Design and Analysis section of the Core Skills modules. This lecture follows on from one pre-recorded session, which was lecture 2, and the original introductory session, which was live. So hopefully you've seen both of those so far. So the job today is to talk about the experimental design part of this part of the course, really focusing on how we design experiments and other kinds of data collection exercises in science, as is probably really nicely exemplified by this photograph on the slide here, or painting. What you've got here is Edward Jenner depicted inoculating a small boy with some pus from a milkmaid's wounds, which on the face of it sounds like a pretty gruesome thing to do. But if you know anything about Edward Jenner, you'll be aware that this moment is kind of a crucial part of the story of the development of vaccines. And today, probably more than ever, we're aware of how important vaccination is as a potential way of solving many of humankind's biggest problems. So in this case, the idea was Jenner had seen that milkmaids seemed to be relatively immune to smallpox, which was a massive problem right into the 20th century before it was finally actually eradicated as a result of vaccination. A deadly disease, but somehow milkmaids seemed less likely to succumb to it. And Jenner reasoned that this might be because they tended to catch cowpox, a closely related viral disease, from cattle, and that somehow having cowpox might prevent you from getting smallpox. So to test this hypothesis, he took some material from an infected wound and injected it into a child, and then gave the child smallpox to see what would happen, which has a number of ethical problems. It also has some technical experimental design problems, which you might be able to reflect on once I've given you this lecture. But in terms of a sort of a seminal experiment representing some of the key components of a good experiment, this was a really good example, and of course it led ultimately to the development of vaccination as we know it today, because thankfully for the boy concerned, he didn't succumb to smallpox, presumably because he was protected by the cowpox vaccination. Okay, that's a bit of a sidetrack really. Let's look at what we're going to deal with today. So it's going to be a three-part lecture as normal. In the first part, I'm going to deal with how we take measurements, how we collect data, the very beginning of the scientific method. We're going to go back to that kind of pastiche of the scientific method and look at the sort of, I say the very beginning, sorry, the central part of the scientific method where we've generated a hypothesis and now we want to test it by collecting data. And in order to gather data, we need normally to take measurements. And measurements are subject to both inaccuracy and imprecision, and we're going to learn about what those two things mean and how they're distinguished, and some ways of combating the problems associated with them. And then we're also going to look at a really important topic called confounding effects or confounding variables, and these are really tricky problems in experimental design, often fatal flaws in experimental design if you don't recognize them in advance. And so I'm going to try to help you explain, understand what a confounding variable is and how potentially to avoid building them into your experimental design. So these are really, together, these two things, key components in our ability to discriminate between the null and the alternative hypothesis, and that's the main learning outcome for the first part of the lecture. Then later on in the lecture, I'm going to talk about statistical power, which is another thing that affects our ability to discriminate between the null and the alternative hypothesis. And although today we're not going to deal with any stats at all, thinking about statistical power is really important ahead of doing an experiment or a set of observations, even when you don't know what the data look like and you're not quite sure what statistical test you might use. It's still important to think about the power of your experiment to tell you something useful. And then towards the end, in the final part of the lecture, I'm going to talk a little bit about biological realism, in other words, the extent to which the experiments and observations that you're making reflect biological reality and are therefore useful in generalizing about the world around us. And finally, I'm going to look at a key distinction between two types of study that we can perform in science: observation and experiment. So I'm going to use a specific definition of the term experiment, as you'll see in a moment. and you'll see that there are different pros and cons to the experimental versus the observational approach, and that's the final learning outcome for today's lecture, is to understand those differences. So here we are with our sort of linear stereotypical view of how the scientific method progresses in the modern scientific world, which I described to you in the first lecture, and we're going to kind of work our way through this scientific method, looking at issues along the way. And we're going to look at this central part of the process where we have a hypothesis, and we're going to use that hypothesis to hopefully generate predictions which can be tested in either experiments or observations. And those experiments and observations need to generate data which we can then use to evaluate the hypothesis, with the potential to reject poor hypotheses and retain good hypotheses. That's the sort of central part of the scientific process that I described to you in the first lecture. By the way, these random footprints are fossilized dinosaur footprints. I'm always struggling for good images to illustrate what's going on in these lectures, and sometimes I just put in things which I think are cool, which in this case, I mean, it's a kind of linear trail of footsteps, which is supposed to be metaphorically equivalent to the scientific process, but it is just cool that dinosaur footprints are still out there for us to look at. Anyway, that was not relevant to the lecture. Okay, so I want to look at this process where we go from having a hypothesis through to having some kind of test of that hypothesis. So typically we're expecting to start with a null hypothesis and an alternative hypothesis. So the null hypothesis says that nothing's happening, there is no difference, there is no trend, whereas the alternative hypothesis says there is a difference or a trend that is of some interest to us as life scientists. Often that hypothesis comes in the form of a prediction, or it immediately can be used to make predictions, and we want to test those predictions using data. So broadly speaking, there are two ways to test a prediction, either using an experiment or by observing the natural world around us. And I'm going to make this distinction that these are two different ways of collecting data, but there is a bit of a grey area here, and you'll find that in different parts of science, particularly the word experiment gets used differently by different people. So to try and explain what I mean, hang on, I think my computer's frozen. There we go. If we look at the distinction between, or the way that I would make the distinction between an observation and an experiment, well, an observation is, once you've got your prediction, you go out into the biological world and collect data which describe that, the part of the biological world that you're trying to study. And you look for patterns in those data which match the predictions you've made. So this is a passive process of observing natural systems doing their natural thing, whether that be, looking down a microscope at some cellular process, or going out into the field and observing animals behaving. What you're doing is you're observing what happens naturally and then looking at whether that corresponds with the prediction of your hypothesis. By contrast, an experiment, at least as defined in these lectures, sort of with a relatively narrow definition, is a data collection exercise which happens in the presence of manipulation of the system that you're interested in. So you, as the scientist, manipulate one or more variables that you're interested in while holding everything else constant, and observe how the system responds. So this might be adding a drug to a patient. You're manipulating the levels of the drug, but you're controlling everything else. So the conditions in which the patient is living or is being tested are identical for every individual. The only thing that's different is the variable you're interested in, which is the drug you're applying. And this manipulative experiment involves intervention by the scientist, and what you do is you look for a response to your intervention, and you are asking whether the response matches your predictions. And the distinction here can be relatively subtle, and people often describe what I would call simply observations as experiments, but the distinction is really important because, as you'll see towards the end of this lecture, it determines what we can and can't say about our hypothesis and about the data once we've done the experiment or observation. So I now want to look at some of the characteristics of a good study, whether it be experimental or observational, and sort of try to identify the key properties of good experimental design. And the first key property I want to look at is the ability to discriminate between the null and the alternative hypothesis. There's no use going out and collecting data with which you're trying to test a hypothesis if the data would look the same regardless of whether the null or the alternative hypothesis was correct. So we're looking for experiments which are cleverly designed to make that distinction. And if Edward Jenner had had a control patient who he had given smallpox to, but without first of all giving them the cowpox vaccination, that would have been a well-designed experiment to discriminate between the effects of smallpox on people who've been vaccinated and those who haven't. But of course there are some substantial ethical concerns with that, because he probably would have killed the control patient if he'd done that. Well, on screen here you've got an example of a similar, simply designed experiment to discriminate. If anybody doesn't recognise this person, this is Head & Shoulders Man. So if you look at the advertising material for Head & Shoulders shampoo, which is an anti-dandruff shampoo, you'll often see this kind of image. We appear to have two identical participants in a study designed to look at the effects of Head & Shoulders. The only difference being that the person at the top has been given an ordinary shampoo, and the person at the bottom has been given the wonderful Head & Shoulders, which removes dandruff or prevents you from getting dandruff. And they're so identical, they've even got the same parting and the same comb and the same clothes, etc. And the only difference is the outcome, and the data here are the presence of the white spots on the upper image, which if you look very closely, look suspiciously like they've been photoshopped on, which are allegedly dandruff. And so the conclusion we reach from this image as representative of an experiment is that Head & Shoulders is effective in preventing dandruff because the lower image shows no dandruff. Now, this might make you think twice about the sort of advertising material you see out there, because this is clearly faked data, and it's clearly the same. It's not just the same participant, it's the same image of that person, and they've just kind of photoshopped on some dandruff. But putting that to one side, if it was a genuine experiment in which you took near identical participants in identical conditions, and the only difference was one of them received Head & Shoulders and one didn't, then that would be quite a good discriminatory experiment where you could discriminate between the null hypothesis that Head & Shoulders has no effect on dandruff and the alternative hypothesis that it does prevent you from getting it. So ideally, I mean, this looks like it demonstrates some of the properties of a discriminatory experiment. But there are some key issues that we need to think about when we drill down into the detail of experimental design, which will affect your ability to discriminate. And I want to look at several of those. First of all, let's look at measurement. So if you're going to collect data, you are effectively measuring something, whether it be on a continuous scale or on a categorical scale. So let's imagine a situation where we're trying to measure the lengths of geckos. It's the sort of thing that ecologists like myself do routinely. Geckos are kind of small to medium-sized lizards, and we might be interested in a whole variety of different things that affect the length of a lizard. But in this case, let's imagine that we're interested in whether there is sexual dimorphism, whether male lizards are different in length on average from female lizards. So we collect a bunch of lizards and we decide to measure their body length from the tip of their snout to the tip of their tail using a measuring device. And the one that comes to hand is a 30 centimetre ruler. And these lizards are less than 30 centimetres long. So this seems like quite a sensible decision. And we're going to measure the length, body length, in millimetres. So that is the level of precision that we've decided to use. The unit that you decide to measure in determines how precise your measurements will be. Millimetres seems like quite a sensible choice for a gecko. We could, however, have chosen a different unit of measurement, a different level of precision. So we could have chosen to measure those lizards to the nearest metre. Now, if all of your geckos are less than 50 centimetres long, then every single gecko will end up with the same measurement being 0 metres long. And as you can see, that's not very useful, because whatever differences there might be in your lizards between males and females, you're not going to pick it up because the precision of your measurements would not be adequate. On the other hand, you might choose to measure the length of the lizard to the nearest thousandth of a millimetre. That would also not be very useful because, you'd gain a lot of extra information, those thousandths of millimetres, but most of the information that you gained would actually be random noise or measurement error, because I don't know if you've ever tried to measure a lizard, but they're quite wriggly things, and it's quite difficult to get the tail lined up in the same consistent way with each individual. There's bound to be some error there caused by the subtle variation from measurement to measurement. Even if you measured the same lizard 10 times, you'd get slightly different results, certainly at the level of a thousandth of a millimetre. So that level of precision would be unwarranted. It wouldn't tell us anything useful. So choosing the sort of Goldilocks level of precision, which allows you to take quick, efficient measurements and yet which gives you meaningful variation in your data set that you can make sense of, is a really important part of making of discriminating in an experiment. But there's another component of oh yes, I meant to say before, imagine now if you had a different scenario where you had the same group of lizards, geckos, and you were interested in comparing the sizes of the eyes of males and females, then suddenly, because the biological problem has changed, the appropriate precision has changed too. So now measuring the size of the eye, the diameter of the eye of a gecko in millimeters, might give you the same result for pretty much every gecko, because it would be 2 or 3 millimeters in diameter presumably, and not very variable because you need more precision. So in this case we might need to use a much more precise measuring device such as digital calipers like they're pictured on this slide, and we might need to perhaps anaesthetize the gecko in order to get them to sit still so that we can take a measurement meaningfully at that level of precision. So precision is all about understanding what is meaningful in the context of your particular biological problem. But there's another thing to be concerned about, and that is accuracy. And unlike precision, where any error that you have is assumed to be random in terms of which direction it goes in, problems with accuracy result from bias in your measurements. In other words, the measurements you take are biased either up or down, or perhaps biased in one sample group compared to another, and that is really potentially problematic. It could, for example, give you a false result for your experiment where it looks like there's a difference between two groups when in fact there isn't. Or, as we can see in the example on the screen I'm just about to describe, it can lead to really unfortunate consequences. So what you can see on screen here is a section of breast tissue viewed down the microscope, and this is the kind of thing that clinicians or clinical microscopists look at in order to try to diagnose breast cancer. And we might be looking for aberrant cells which show signs of cancer in a sample of otherwise healthy cells, and we might be scanning dozens and dozens of these microscope slides every day in the hope of spotting the one individual who has cancer, who's had a biopsy taken. And you can imagine that bias here in our estimates of the number of aberrant cells which might be cancerous is going to be really important in terms of patient outcomes. If you're a hard-pushed microscopist in the lab, you've been looking at thousands of these things, your concentration starts to wander and you start to systematically under-record the number of aberrant cells, then this would be an example of inaccuracy or bias. Your counts of aberrant cells would be lower than they really are on average. That could lead to missing positive diagnoses of cancer, and therefore, some potentially really horrible consequences. The opposite kind of bias would also be a really big problem. If your mind is sort of prone to wandering imaginatively and thinking that you're seeing aberrant cells when you're not, it might be quite hard to define what is an aberrant looking potentially cancerous cell from what is a healthy looking cell. If you have maybe one of the technicians in the lab is systematically over recording because they're classifying healthy cells as aberrant, then you run the risk of giving a positive diagnosis when in fact there isn't cancer present. So here, bias in our assessment, in our measurement of the thing that we're interested in, could be really critical. I mean, in that case, in terms of clinical outcomes, but more generally in terms of the scientific outcome of an experiment or a set of observations. So those are issues with measurement, but there are other things which affect your ability to discriminate in an experiment or a set of observations. And one really important thing that affects your ability to discriminate, which is really difficult to get your head around, is the concept of confounding variables. So typically we might be interested in the effect of one variable on another, let's say the effect of A on B. And so we're measuring A and B and we're looking for an association between the two. And often we will infer from that there is a causal relationship between the two. Changes in A cause changes in B, or vice versa. The problem is that very often, depending on how we've set up our experiment or observations, there are alternative possibilities. So if we find an association between A and B, when A goes up, B goes up, that might be because A has a relationship with another variable. Let's call it C. A and C might be causally related, and C might have a link to B. And even though there's no direct link between A and B, we could get a relationship between A and B caused entirely by this separate confounding variable C. Now, I've talked it through in abstract terms there, and you might be thinking, what on earth is this about? Let's look at it in some real biological studies, and we'll start to see why this is a real problem. So, first up, let's have a look at this chap here. This is a video of a grasshopper mouse, and it is attacking a scorpion, which for most mice is a pretty stupid thing to do. And in fact, you can see that the mouse has been stung, and yet the mouse, although it looks mildly irritated by the sting, carries on attacking the scorpion until it's subdued, eventually dead, and then it eats it. And that turns out to be what grasshopper mice do. They like to eat scorpions, and amazingly, they seem to be more or less immune from the effects of the sting. And this contrasts with other mammals, including other mice, which if they were stung would either die or be in extreme pain and suffer really badly. So we've got something weird going on here, and this was the subject of a neuroscientific study actually, to try and understand the underlying neuroscience that explains why grasshopper mice are not affected by the venom in the same way that other species are. So imagine we're doing an experiment to compare two species of mice, the grasshopper mouse on the right there and the ordinary domestic mouse that we use in laboratory experiments traditionally in science. Imagine we want to try to compare tissue samples from these species in order to understand whether their neurons respond differently to the venom, and that might explain the difference that we see if the animals get stung. So let's think of a kind of thought experiment. Let's imagine we've got these two species, we want to compare them. So what we'll do is we've got lots of lab mice knocking around, so we'll prepare some samples of their neurological tissue, and then we'll expose it to the venom. We're going to need some venom. Okay, so we'll go and collect a whole load of scorpions from the wild, we'll milk the venom from them, and we'll put that in storage ready for the experiment. So right, we'll now expose all our domestic mouse tissue to the venom, and then we're going to need some grasshopper mice, so we'll go out and do some field work and collect some mice, take some neurological samples, and run the test with the venom on those guys. The problem with the design I've just described to you, you may have spotted, and it's written on the slide here, is what we call an order effect, which is a special example of a confounding effect. There's a confounding variable here, which is time. So I've done all my domestic lab mice first, and then my grasshopper mice later. And for lots of different reasons, the results I get might vary through time, completely independently of what species of mouse that I'm working on. So in this case, for example, if I collected all the venom at the beginning of the study, the venom might have a shelf life. It might start to go off. It's typically proteinaceous. That protein might start to denature or degrade. And by the time we get to sampling the grasshopper mice, perhaps several weeks after we started the study, the venom may have lost all activity. And so if we find lower impact of the venom on the neurons of the grasshopper mouse, that might be simply because the venom's no longer effective. It might be nothing to do with the fact that we're looking at a different species of mouse. So the point I'm making here is that time is a potential confounding effect, a confounding variable. And if we think back to that sort of diagrammatic representation on the earlier slide, we were trying to look for an association between species and neurological activity. Let's call those two things A and B. And there was this intervening variable C, which was time. And time was associated with species because we collected the species at different times, or we used their samples at different times. And time was also associated with the neurological response because of the effect of time on the venom. So in this case, we think we know what the confounding variable is, but in other cases it can be difficult to know what the confounding variable is. And avoiding confounding effects in experimental design is really the single most important problem that you're going to face when designing a good experiment. And I will come on to how we avoid that in a second, or some strategies for avoiding confounding effects in a second. But if we just look at the data from this was a real study actually of these grasshopper mice, because I know you want to know what was going on. Well, it turns out here we've got on the y-axis the number of action potentials in the dorsal root ganglion neuron. So we're looking at a particular type of neuron. We're looking for the number of times those neurons fire when exposed to the venom. And in mus musculus, which is the domestic mouse, as you increase the concentration of the venom, what you see is a massive increase in the number of action potentials. So, and this is showing how the venom acts on the animal. It's triggering a massive neurological response, which can be fatal. But in the grasshopper mouse, in fact, if anything, when you treat those samples with the venom, what you see is a reduction in neurological activity. So the mouse really does seem to be fundamentally sort of immune from the neuroactive properties of this venom. Anyway, that was a side story, really. We're dealing with this problem of the confounding variable. Another way of thinking about this problem is to think of it as a lack of independence between the data points in each of the two samples that we've got. We've got these two sets of samples from two different mice, and we want them to be as independent as possible in everything apart from the thing that we're interested in. And the thing that we're interested in is the species of mouse. So obviously if you take multiple data points from grasshopper mice, they share that in common, they all come from the same species. But we don't want them to also share other things in common. We want them to be independent. If they all come from the same time period, then suddenly they're no longer independent. And similarly, if we'd used two different technicians to help prepare the samples, one for the domestic mice, one for the grasshopper mice, that could have an effect on the results. It could be a confounding variable because it creates a lack of independence among the data points from each species. There are lots of other ways in which you can generate confounding effects which are equally troubling. So here's another example. Imagine that we were spraying a fertilizer onto fields of crop, fields of, let's say, wheat. We're looking to see whether this fertilizer increases plant growth and yields of wheat. So we get our tractor and we load it up with the fertilizer, suspended or dissolved in water as a solvent, and then we spray the fertilizer onto the crop. And we come back several months later and we compare the fields that we sprayed with the fertilizer to a control set of fields that weren't sprayed. And we find, great, the plants have grown much better when they were sprayed with the fertilizer, so we conclude the fertilizer was beneficial. There's a key problem with this design, which is that we had to put the fertilizer in solution and spray it on in an aqueous form to the crop. And what are plants like? Well, they might like fertilizer, but they certainly like water. And if this had been a particularly dry year, perhaps the control crop was suffering from drought and therefore not growing very well. And the addition of the fertilizer had the unwanted beneficial effect that you were irrigating the crop and it was growing better because it had access to more water. So here we thought we were looking at variable A, fertilizer, affecting variable B, plant growth, and in fact there's this confounding effect of variable C, the amount of water available, which was driving the apparent relationship between the two. So another classic kind of problem in experimental design is an unwanted effect of a manipulation. You think you're manipulating only one thing, but actually you're accidentally manipulating other things. So how do you deal with this problem? Well, what you want to try to do as a sort of starting point is to randomize your data within an experimental treatment so that the data are independent as much as possible. So if you think about our mouse study, we would want to randomize the times at which we sampled the mice, the two different species, so that it wasn't that we were taking all of one species first and all of another species later, but that we were taking them at random times throughout the study. And that would solve any problem of the venom going off over time, because any effect of that degradation of the venom would not affect one species more than the other, and so we'd still be able to detect the effect we're interested in. So randomization often helps. Sometimes you can't do that, so in the case of our grasshopper mouse study, we might be limited in when we can get hold of grasshopper mice. Perhaps we only go out into the field on every second Friday or something, and so every few weeks we get a few grasshopper mice in. If that's the case, then another alternative strategy is to measure the confounding variable that we're worried about and include it in the analysis. So in this case, we could measure the time at which we took each sample and put that in as a predictor in the analysis, and essentially remove the effect of time and then look to see if there's still a remaining effect of species on top of that temporal effect on the neurological activity that we were observing. In the case of unwanted effects of manipulation, nearly always the solution is to design your manipulation carefully enough and your controls carefully enough to mean that the difference between treated and control individuals, in this case it's fields, is only the variable you're interested in. So in our case, there's an easy solution here. We can add water to the control crop without the fertilizer in solution, so that the two crops experience the same conditions with the exception only of the addition of the fertilizer. So in other words, we just design the manipulation better and the controls are more similar to the manipulated fields, in fact identical in every respect except for the fact that they had fertilizer added to one and not the other. Okay, so that's the end of the first section. After the break we're going to go on to look at statistical power and biological realism.